Okay, so this is my last lecture, and again, I want to thank everybody for coming, and especially after other many hours of lectures. And I think, again, I'm very grateful to the organizers, and I think it's very important that we keep science going during these difficult times. And I think we're probably pretty fortunate to have technology that lets me amazingly talk to you and listen to your questions on the other side of the world. I think that's quite a nice achievement, but I would much rather meet you all in person. So maybe I'll be able to do that someday. In any event, for those who are interested, I just put a few references here for the first two lectures. So here's a review article by the lead author is Kirill Korolev about spatial population genetics and the Ito calculus and some of these experiments, inspired by some of these experiments that I've told you about. The work on pushed genetic waves and antagonistic interactions especially it came about in collaboration with Hidenori Tanaka. There's this paper. We were actually studying a problem with antagonism associated with CRISPR-Cas9 and trying to figure out if some CRISPR modified organism, if it escaped from the laboratory, how fast would it spread and stuff like that. And it turns out it can spread by either a pushed or pulled genetic wave. And then there's this paper I mentioned yesterday by Max Levenchevich and myself on nucleation of droplets in the presence of these number fluctuations. So today I'm going to, I'll briefly revisit this antagonistic pushed wave problem, but then I'll move on to talk about microbial interactions and range expansions on liquid substrates. And that produces sometimes pictures like what you see here at the lower right. This is a yeast colony floating on a very viscous but still liquid agar medium in a Petri dish. So again, you can see the Petri dish curved walls out here, but it has a very different range expansion than on a solid agar substrate, a hard agar substrate. And in fact, it generates its own fluid mechanics. It generates, it turns out, a submerged vortex ring. And it took us a couple years to sort of understand this and think about it, we hope in a useful way. But I hope you'll find these problems where we have a marriage between fluid mechanics and spatial population genetics interesting. And that's the topic of this last lecture. And again, thanks to the organizers for having me. And here are two papers that will cover in more detail what I'll talk about. One is the experiments by Severine Addis and collaborators are in this PhysRev X paper. And then we just posted a preprint with my collaborators, including Suraj Shankar, on the web of fluid mechanics and spatial population genetics, mostly with antagonism. And as always, I welcome experts from, I welcome questions from experts like Kavita, but especially welcome questions from the students and postdocs, and please keep those questions coming as we proceed. So this is just to finish up some things that we touched on last time with these waves and Fisher waves and various other kinds of waves. This is a simulation of a Fisher wave going through an array of obstacles. And so you'll see it happening up here. Here's a wave of expansion. We're just solving that partial differential equation that we talked about on Wednesday originally. And This is the wave of advance. It could be a new genotype, could be just a population expanding across virgin territory. And these red diamonds are regions where the growth rate is 0. So you can think of these as, they could be like lakes in a landscape, periodic array of lakes in a landscape for organisms that move over the land, so that they can diffuse in these red diamonds, but they can't, they're not growing. So you eliminate one of the terms, two of the terms in the Fisher equation that correspond to growth and saturation in these red diamonds, but otherwise it goes. And well, you can see that not much happens. The population envelops these red diamonds, and there's some slow change within the red diamonds because there's diffusion of organisms in there, but they're not reproducing. So not much happens here. There are little cusps that you'll see downstream of the diamonds. You can see a formation of a cusp. So you can actually understand this using a variant of geometrical optics. And these cusps downstream of the diamonds are a little bit like, you can use ray optics actually to understand them. They're caustics in the pattern here. It's not interference per se, but there are cusps. The cusps heal, and otherwise, though, you can see the overall population, the overall propagation velocity is unchanged. Very hard to stop Fisher waves with barriers. In fact, it's very hard to stop almost any pulled wave with barriers. But here's, again, Fisher wave doing the same thing, but now the diamonds are closer together. So if we look at what happens now, let me stop this temporarily. This front here is where the Fisher wave would be if there were no diamonds. But this turquoise region here is where the actual population is. So the blue is just a marker as to where a population would be. And you can see that the wave is slowing down. Now you could say it's sort of slowing down in a trivial fashion because it has to zigzag its way to go from left to right. And so it takes longer for a given velocity. But that's actually a kind of interesting feature of these media with complex arrays of obstacles. They have a kind of index of refraction, just like the speed of light slows down in window glass, and we get bending of beams of light due to refraction. Something similar happens here, and you can actually see these population waves bend. These Fisher waves bend if you set up the simulation, and we've also done experiments in the right way. There's an experimental paper by Wolfram Möbius, myself, and Andrew Murray. But these are pulled waves. These are the conventional ones with a simple quadratic nonlinearity in the reaction-diffusion equation. But pushed waves are much more fragile. We already saw that in a sense because we saw that to launch a pushed wave, even if there's a selective advantage driving it in some genetic population, had to be larger than a particular size. And so here's just a reminder of the kind of equation that has a cubic over here with some sort of fixed point value. And this is what comes about when we have antagonism. Sigma is the antagonism parameter. Delta was that selective advantage that we talked about. And F is the concentration of a particular genotype. And if I do a simulation for a certain parameter regime where I get rid of this F star, or it's unimportant, say F star is outside the range 0 to 1, so there's no extra fixed point, then it turns out that this is a pulled wave, and here's an array, here's a barrier where we have basically lethal region That's what 1.0 means, it turns out. And we have a gap. And this pulled Fisher wave without the cubic polynomial, just quadratic terms, say, and also similar polynomials that don't have a relevant F star in the range of interest, it goes right through. It goes right through the gap, just like you saw in that earlier movie. However, if I change parameters and I make this F star activate, and that's what I'm doing with this thing called S. It's not exactly a selective advantage, but it lets us tune the polynomial on the right. You can see that, in fact, this advancing genotype coming into the wild type domain reaches this gap, and it doesn't go any further. It just stops. And roughly speaking, this is called the Ali effect in population dynamics. And it stops, roughly speaking, it stops because the gap is smaller than the size needed to nucleate a pushed wave. So these are pushed waves where the dynamics is controlled further back rather than in the foot of the wave. So I just wanted to follow up on that. So any questions on these pushed and pulled waves before I move on to talk about fluid mechanics? Okay. Yes, please, preach.
Yeah, just a small question. Like in this A case, case
Yep.
A, just near the barrier, won't there be like some kind of like if you think as a river flowing, so there should be some circulations, right? Like small.
Well, okay, so if this were a liquid medium, there could certainly be vortices, backflow, and so forth. But from that perspective, we're working in a very overdamped region and regime, an overdamped regime. And so, although I will be talking about backflow a lot in a minute, in genuine fluid medium, here, imagine it's going across a hard agar substrate. So we don't have inertia and backflow in this equation, but we will have it in 1 or 2 slides. So thank you for the question.
Yeah, so maybe I misunderstood the point, but for the pushed wave, so nucleation hasn't happened before. I mean, that hasn't happened before 40 time steps or so.
Okay, so thank you for the question, and I also saw another raised hand. So anyway, to answer that question, nucleation has already happened, say down here, at t equals 0. So what I have is a planar front of a favorable genotype coming forward and hitting a barrier which has a hole in it. And so the purple is where the organism can't grow at all. It dies if it goes in there. But once it hits a gap, if the gap is small enough, it won't make it through. And that's what's happening here.
I'm just thinking of the usual StatMech. I mean, once nucleation happens, the domain coarsening and all that stuff will just grow.
Sure.
Once the nucleation has happened. So I'm like confused here. If nucleation has already happened, then why is this barrier not letting?
Yeah, so that's a very, very good point. Nucleation has happened and we're arresting it. And that's, I think, a consequence of the fact that this is not literally like the nucleation that we're both familiar with in physical chemistry, nucleation of droplets of water vapor from a gas or something like that. This is this nucleation phenomena within the framework of population genetics. And the reason why it stops is that this gap is smaller than the critical radius for growth. You can check that if the gap is small enough, it doesn't get through. If it's bigger than that critical nucleus, then it can get through. And I find this picture striking every time I look at it because it goes so counter to our intuition. But that's what happens with this equation.
So I should not just think of the usual conventional nucleation phenomenon. So there's some differences. Is that
it's not literally the same. Exactly. There are some differences, and one of the most important differences is that the noise, the fluctuations, only happen at the interface. I think I mentioned this last time. It's as if the interface itself between this genotype and the wild type, we call it the gene drive here, but this genotype and the wild type, it's as if that interface is hot, but the rest of it is at very low temperature. And so getting the nuclei that you're interested in it isn't possible. So it's as if this wild type, if it were a stat mech system, was down at 0 temperature. This is down at 0 temperature. It's only heated along this rim. And when under those circumstances, it has to be bigger than a certain size to spread. And this gap is smaller than that critical size. So it is a very counterintuitive, striking phenomenon.
Yeah, thank you.
Yeah, and was there another question? Was someone else raised their hand?
Yes, sir.
Yes, please.
If I am not wrong, that S is the selection coefficient, right?
It is something like a selection coefficient, but the parameters sigma and F depend on it in a complicated way. This picture arose. It's actually a selective disadvantage. It's the selective disadvantage that a gene drive has, but by virtue of the way it was constructed with genetic engineering. I don't remember all details, so I probably shouldn't go into them in the interest of time anyway, but it's not a conventional selective advantage. It's more like a selective disadvantage for the gene drive. One of the amazing things about these gene drives, if you know about them, is that even if they have a selective disadvantage, they come in, they arise, they can be engineered to arise in sexually reproducing organisms, like mosquitoes. And even if the mosquito with the gene drive doesn't grow as fast, doesn't reproduce as much, in a fertilized egg, what happens is, if you have a big A, little a heterozygote in a fertilized egg, the gene drive attacks, suppose it's called capital A, it attacks the little a chromosome and replaces it, little a chromosome gets replaced with big A copy. So because of that, it still spreads, even though it has a selective disadvantage, which we're parameterizing by S. But that's another story. But you're right, it's in the spirit of the selective advantage that I was talking about before, but it's a peculiar kind of S that people use in the context of gene drives. So I saw there's another hand up, I thought.
Yeah, hi sir. I just wanted to know what was exactly the biological meaning of the two barriers, which say one written on.
Okay, yeah, good, thank you. So the biological medium, thank you for the question. The biological medium might be, if these were, suppose this was a mosquito breed that it doesn't carry malaria, and you wanted to replace the wild type in the white area, which does carry malaria, but maybe it accidentally got out of the lab and you didn't want it to spread, you wanted to stop the experiment where it takes over the wild type. Then these purple barriers could be an insecticide that you lay down in the environment, in the two-dimensional environment, where you're doing this hopefully controlled experiment that causes any mosquitoes that go in there to die, either wild type or the gene drive. So that's what so imagine you laid down a strip, or this could be a forest fire, and now these are fire breaks in the forest fire, and then although in that case it would probably be a pull fissure wave, but there would be fire breaks with no material that the fire can consume, and that would be another alternative meaning of the purple regions. I hope that answers the question.
Yeah, thank you.
You're welcome. Okay, so that's just finishing up from yesterday. But now I want to talk about population genetics and fluid mechanics. So I'm changing the focus here. The motivation is that life, as you probably know, very likely evolved in a liquid environment, not on a hard agar plate, not on the surface of the earth. Life came out of the oceans, eventually, and colonized the surface of the earth. But 2 to 3 billion years ago, also lots of water around. And there's evidence in the fossil record for an amazing kind of bacteria. Here's an example of modern day equivalent that they're green, and they're green because they invented photosynthesis. They invented photosynthesis 2 billion years ago. The same photosynthesis we see in plants and algae, for example. Photosynthesis has as its byproduct, the waste product, oxygen. And that changed the Earth dramatically, maybe even more dramatically than we humans are changing the Earth now with worries about global warming. These cyanobacteria produce oxygen as a waste product, and that's why we have oxygen in our atmosphere. And of course, that eventually, although it killed off a lot of other organisms for which oxygen was a poison. It's a little bit like the toxins that I was talking about earlier. They produced oxygen, which was a toxin, not to them, but to other organisms. Evolution eventually found a way to exploit the oxygen and produce very energy efficient multicellular organisms like ourselves that use oxygen to make their own energy. But this is an organism that takes light, takes photons, harvests photons, and makes its energy that way. And their spatial growth and evolutionary competition took place in the ocean. They're still in the oceans today. And if those of you who have had a course in fluid mechanics remember the concept of Reynolds number, that tells you how agitated the fluid is. This can happen both at high Reynolds numbers where the fluid is very agitated and at low Reynolds numbers as well. And these organisms, very interestingly, were absorbed into other more complicated eukaryotic organisms, and they became the chloroplasts of green plants. And they also are, so they're now parasites, if you like, or symbiotically attached with green plants, and that's how green plants, of course, get their energy. But it happened first in bacteria, and then plankton, which are more complicated than bacteria, but they have also absorbed the ancient ancestors of these cyanobacteria. This particular one is called Senecacoccus, and that's how they harvest light by using these symbiotic organisms. And these photosynthetic organisms, they can't exist at all heights in the ocean, because if they go too deep, there are no photons down there. The light is all absorbed. So they tend to control their height. They actually control their buoyancy to resist possible downwelling currents, to stay close to an ocean or lake surface. And in fact, here's a example of a cyanobacterial bloom. This bloom is happening on a lake in Guatemala. You see these beautiful green striations, which are taking place at the surface of this lake. Not literally at the surface, but maybe in the first 50 meters of depth, where you can still get light in significant concentrations. And it sort of looks like the early stages of mixing cream into your coffee, perhaps. But I want to convince you it's actually fundamentally different. These striations, at least if there's some agitation in the convective flow of the water in the lake, will never become uniform the way cream becomes uniformly dispersed in your coffee. And I'll try to show you why that is. Here's some other pictures of phytoplankton blooms. These are some of these little organisms that can live in the, say, the North Sea or the Atlantic Ocean, the Pacific Ocean, the Indian Ocean, whatever. So here's a plankton bloom in the Norwegian Sea. Notice the scale is quite large. It's 110 kilometers. These are not experiments done in a Petri dish. This is off the southern coast of Iceland. And you see these very complicated striations. It looks like a little bit like a famous problem in turbulence called diffusion of a passive scalar. But in fact, it's not like that for a number of reasons, but one of the reasons is that these are alive. And they have a reproduction time, not 20 minutes, not 40 minutes like the bacteria we study in the lab, but maybe 12 to 24 hours would be how long they double. But once you have this a turbulent ocean, and there's lots of sources of turbulence in the ocean, which I'll talk about, you have eddies of all different scales. That's related to the Kolmogorov theory of turbulence. And one of my collaborators, Prasad Perlikar, knows a lot about turbulence, and I'll show you some of his simulations. But there are eddies of all different sizes, and you might imagine that if these are growing, it's important to know what that doubling time is relative to eddy turnover time. How long does it take for a fluid particle trapped in an eddy to go around 360 degrees? And since there are many eddies, many different size eddies in oceanic turbulence, then there are many eddy turnover times, and they range from a few minutes to 50 days out there in the ocean. And this plankton doubling time is right in the middle of this range, this very large range of times. And so some of the eddies for the plankton are very fast, and they can sort of average over them because they don't double significantly as you go around in the eddy. But other eddy turnover times are very slow compared to the doubling time. And then the fluid mechanics can have an important effect on what's going on. So here's another, I think, quite beautiful picture from satellite imaging off the coast of South America. So I think these are the Falkland Islands, and this is Argentina over here. Then this is a chlorophyll map. It's from the satellites, maybe the reflection of the sun or something. You can actually get the density of chlorophyll. That, of course, is a sign that there is oceanic life here. And here are the corresponding fluid contours. See, these are the streamlines, if you like, associated with the South Atlantic Ocean. And you get this from the height. You can infer assuming incompressibility of the ocean, you can infer a kind of streamline. This is some analog, I guess, of the Gulf Stream off the coast of South America. But you can also look at more precisely at the type of chlorophyll that's coming back and look at the dominant species types that are in here, and they're different colors here, different types of organisms as identified by subtle variations in their chlorophyll. And there's some, like this particular aqua thing seems to track this particular feature in the fluid flow. So this is just to show you that there's a kind of ecological seascape associated with these flows when I have reproducing organisms that are competing with each other, possibly with antagonism, maybe with mutualism, with Darwinian selective advantages. And so what I've, with my collaborators, been trying to do for about 10 years is to arrange a kind of shotgun, we call it a shotgun wedding of these two fields of fluid mechanics and spatial population genetics. So this is one of the motivations. And how do you describe something like this? Let's imagine we just have one species type. So it'll be like our Fisher equation problem that we started these lectures with. And C of X and T would be, say, some average two-dimensional density of organisms integrating over the first 50 meters of the oceanic surface. And so, of course, there's the Navier-Stokes equations and stuff like that, and that's going to produce some advecting fluid velocity here. But if I neglect this advection term, this is just the Fisher equation that I was talking about. I happen to have rescaled the population so that the carrying capacity is 1, and now I have a growth rate which is mu out in front instead of A. But otherwise, if I neglect this term, this is the equation that will have a Fisher wave. But now I want to couple it to fluid mechanics and put in an advecting velocity field. And you can get this velocity field by just solving the three-dimensional Navier-Stokes equations and looking at what happens near the surface if you wish. But it turns out, and I'll show you why in a minute, that unlike most fluid mechanics problems that you might have encountered, this is effectively if it's a 2D, if this is a 2D population that's somehow localizing itself near the surface of the ocean, the effective 2D velocity field is compressible. It has a non-zero divergence. And that changes the physics dramatically, as does the growth compared to diffusion of a passive scalar, for example. So here's the justification for that. This ocean is being stirred by Langmuir's circulation cells, for example, and by heating of the water and so forth. And of course, there's three-dimensional, virtually incompressible flows going on here. But if the organisms, and here's a little cartoon, don't go down, if they control their buoyancy so that they don't go down below 30 meters or so, which they almost have to do so they can get the benefit of the photosynthetic light, then they'll accumulate at these downwellings. And if I coarse grain a little bit to think about effective 2D velocity field, it's going to have a non-zero divergence. And that will make the population generically inhomogeneous when we have competing genotypes, for example. In a simple Fisher equation without this advection, as we saw, the system is trying to grow up to a population density of 1 in this rescaled unit, and it'll just be 1 everywhere. And that can't happen if you have this buoyancy and effectively 2D compressible turbulence. And just to get a sense of how that changes things, here's this beautiful simulation by Prasad Parlikar and his collaborators, which just, it's a color map of the density of organisms, where what Prasad did was to solve the Navier-Stokes equations in 3D, and I think it was in a 3D cube with periodic boundary conditions and some very high Reynolds number, like 100,000. And then he had advection and diffusion, growth, and saturation, just what we had in our original Fisher equation, but with the addition of advection in a 2D cut through the cube. And so the idea is the organisms in this little model would want to concentrate on the midplane of the cube. And so this is showing the concentration of organisms on that midplane or on a very thin plane where you just take the 3D velocity field and you project it and only allow it to act on this 2D plane. And you can see this is, I think, quite a remarkable simulation because the organisms never die out, but they get concentrated. They get concentrated by this non-zero divergence flow. And when they get concentrated larger than the carrying capacity, so they get concentration effectively gets greater than 1, then the effective growth rate from this nonlinear term is negative. And so the population crashes. So the effect of this 2D.
Sorry, David, I missed it.
Please.
So the green one is the population density or white? I mean, I'm not sure.
So thank you. Thank you for the question, Kavita. I didn't make that clear. It's a color map. And so of a single species. And so white is nothing there. Green is a particularly high intensity and blue is some intermediate intensity. So it's just a way of representing, if you think of it like topographic contours of population density, but we just took a set of contours and above a certain height, colored those green, intermediate height, color those blue, and below a certain threshold, it's white. And to first approximation, there are no organisms there. I hope that.
Right. Thank you.
Thank you. So you can have a lot of fun, I think, at least my collaborators and I thought so, playing around with various dimensionless parameters that are much beloved by fluid mechanicians, like the Reynolds number. That's the typical velocity times the size of the system divided by the shear viscosity of the liquid. There's something called the Schmidt number, which is a ratio of this shear viscosity that comes into the Navier-Stokes equations, and the diffusion constant that tells you how these organisms are able to move around. So maybe they have flagella and there's some run and tumble effective diffusion constant. But there's a new number here, which is a ratio of a doubling time of the organisms. That's tau 2. That's basically 1/mu. So that's how long it takes, if there were a very dilute concentration of organisms, that's how long it takes for them to grow up. And then you can then look at a typical eddy turnover time for the flow, say a small eddy turnover time. And then the ratio of these numbers, which is also 1/mu times the eddy turnover time, that's another dimensionless number. And the interesting biology happens when the eddy turnover times of interest are long compared to the doubling time. Because then as one of these organisms is advecting around, here's a kind of an eddy that's happening, it's drifting as well. As the organism is advecting around, it can grow up and change and respond. So that's one genotype only. What happens if I have more than one? So here's another beautiful simulation from Prasad, which basically is a agent-based simulation where we have two interacting species. I'll just show you the beginning here, and I'll tell you what's going to happen. We have green organisms and red organisms subject to that advecting velocity field. Kappa is a measure here of the degree of compressibility, which Prasad can control. And these are, if you like, the Feynman diagrams of the organisms. Here's a red Seneca-coccus cyanobacteria, let's say, with a red label, just like in those agar plate experiments. And here it is undergoing cell division. Okay, so this is a space-time plot of what's happening with these cells. Here's a green one. Okay, and then there's another process that occurs in this agent-based simulation, which is in this case going to be an example of survival of the luckiest. These are neutral In this case, they don't have to be, but we'll take them to be neutral. And so black here is another process which black could be either red or green. If the concentration gets too high locally, there's not enough nutrient, and so the organisms die. And if you coarse grain this kind of thing, you'll get Fisher genetic equations, Fisher population equations, and things like that. So, but it's an agent-based simulation. So let's watch what happens. So I start off with uniform mixing of the red and green species in 50-50 concentrations. They are subject to this compressible turbulence. And well, you can see that it's a disaster in a sense for the organisms because they get squeezed down by the non-zero divergence of the advecting velocity field, just like what we saw on the previous slide. This is just what we saw here, but in this case, it's a even more dramatic effect. But the organisms don't die out; they just get very dilute. So the turbulence, the compressible turbulence, reduces the carrying capacity dramatically. You can see little eddies going on here. Let me go back a bit. Here's an eddy, for example. And if you look carefully, you can see that these green organisms are dividing sometimes they go away. So the red organisms are dividing, sometimes they go away if by this black process here. So maybe I can ask this audience, you don't have to actually raise your hand, but you might want to think to yourself, which organism do you think will win? If I go long enough, you might expect that red or green will go to fixation. And most audiences, when I asked them this, at least before COVID, and people could easily raise their hands, would say green. But in fact, if you go long enough, I'll just take it out really far here. See if I can do it. It's red. But I can assure you that if you do this experiment over and over again, half the time red wins and half the time green wins. Okay, so there's a randomness, stochasticity to this, which has superimposed on the stochasticity of the fluid mechanics. So it's a very rich problem. And so for a while, my collaborators and I have been interested in trying to find simplified model systems. We're not going to explain the ocean overnight or even in a month or two, but simplified models that will help us try to understand what's going on. So we want simplified models or possibly experiments that explore how fluid flows affect the spatial population genetics that I've been telling you about in the first two lectures. And so high Reynolds number, maybe we can't get that in the laboratory so easily, but low Reynolds numbers are also biologically relevant. And as I said, the interesting regime is a regime where we impose flows so that there are reproduction times, doubling times, the analog of 20 minutes or 40 minutes for a bacteria, that are much smaller than the eddy turnover time. So as one of these organisms is infected around, it has time to reproduce and respond to whatever the flow is doing. So that's been a goal, and I think we've sort of made some progress. I hope to convince you of that. But before I go on, maybe I should pause and ask if there are any questions on the motivation or the background for the rest of this presentation. So let me know if you have any questions at this point. Here, I'll back this up so you can see this amazing collapse that Prasad discovered. Okay, if I'm not hearing any questions, I will go on. And now this is a more general model in the sense that it answers one of the questions that was asked last time about two competing organisms, where is the regime of exponential growth? And so here I have a set of two coupled reaction diffusion problems. Let's leave out this advecting field for the moment. And this is just a rewriting in some sense of what I talked about in the first and second lectures. There's two species, A and B, or it could be red and green perhaps. And then there's these nonlinear terms in the reaction part here. There's diffusion constants for both of them. And then the nonlinear terms have the following structure. If I didn't have these epsilons, suppose the epsilons were 0, then the whole population would just grow up until CA plus CB was 1 in this model. And that would be true for both populations. So when both populations are saturating so that CA plus CB is equal to 1, that's where I might want to look for interesting Fisher population waves, Fisher genetic waves, and that sort of thing, especially the genetic waves. But now I'm going to perturb that simple model just a bit by adding in some epsilons here. And these epsilons are going to change this effective growth rate in parentheses here. They're going to change it by tweaking it with either positive or negative value. That could be mutualism if the epsilons are positive, or it could be antagonism if they're negative. And these epsilons are nothing more than a simple generalization of the alpha and beta parameters of the talk from yesterday. But I want to make it a little more interesting than that. This model, as stated for now, will have exponential growth. I've also chosen units of time so that the growth rate is 1, but there should be maybe an A or a mu parameter out in front here. But there'll be exponential growth in the early stages, and then it can grow up, and then maybe once it grows up so that CA plus CB is approximately 1, then there can be small modifications if these epsilons are small that reflect mutualism, Darwinian selective advantage, antagonism, etc. To make the problem more interesting, though, we also added advection. And the advection could be compressible, it could be incompressible. And rather than to put in a full turbulent advecting field, we just put in a simple cellular flow. And again, in the early, the first paper on this Prasad Perlikar was very much involved in this. And here's the velocity field that we put in. It has an overall strength, capital F. And then there's a parameter alpha. And as you tune this parameter, as you'll see, you can make the flow have sources and sinks, and hence it'll be compressible. You can make it have saddle points and all sorts of interesting structure. And we want to see how this flow is going to affect the survival of the luckiest, the Darwinian competition between the genotypes, etc. And here are some of the typical parameters in the simulations that I'll be showing you. D is a dimensionless diffusion constant. L is the size of the system. Alpha equals 0 corresponds, I think, to a sink in this flow field. And then the epsilons will be negative, but not necessarily equal, reflecting antagonism.
I have a question.
Sure.
Yeah, so just forgetting the flow part, and if
Yeah.
I had epsilon A and epsilon B to be 0, then I can see that CA plus CB basically obeys the usual Fisher genetic equation, and we have
That's
just a filled wave. But now when you have epsilons, then what happens? Which one, I mean, both will be pushed, both are pulled, or like, can you say a bit more?
You'll typically, depends. So I'll tell you the answer briefly, and then I'll show you how you derive it in the next slide. So if suppose epsilon A and epsilon B are opposite in sign. So one organism helps the other, but is hurt by its counterpart. Then there'll be, in fact, a pulled wave. But if epsilon A and epsilon B are negative.
But pull wave for which one? Like there are two concentrations. So which one is the one that?
Okay, so it's good. So I think the best way is to go to the next slide. So if you'll bear with me, I'll show you how we make contact with exactly your question. So let me just do that if I can. And I won't go through all this in detail because I want to make sure I cover a certain things. But here's the idea, Kavita. So here is a, see if I can find my marker. There it is. These are the equations we were just talking about up here. And now, if the system grows up so that CA plus CB is approximately 1, then the remaining biology or the remaining science will come from the epsilon A's and epsilon B's. And so the way to analyze this mathematically is to define two new variables, CA plus CB, and then this fraction F that is exactly the F I was talking about in the context of push and pulled waves. It's the fraction of the A species. So this is CA over the total. So you take these equations, you change these variables, and look at the system in terms of these new variables. And the first thing you can look at is what is the dynamics of the sum of these things. And to get these pushed and pulled waves, Kavita, I'm going to want that sum to be close to 1. Doesn't have to be exactly 1, but close to 1. So let's look at this thing. If CT is close to 1, then this term will be small. But I still need to look at this term here. And if I just neglect diffusion, for example, just to get a sense of what's happening locally in space, when CT is 1, then this time derivative thing, sigma epsilon A and epsilon B are negative, so the sigma parameter is positive, and what's happening locally at interfaces, so F times 1 minus F, it's when both fractions are present at, say, concentration roughly a half. If I'm away from interface, this is 0, and so it stays 1. But at interfaces, it dips down, and it dips down at a rate given by this antagonism parameter. If you imagine that these two species are at war, Kavita, this is like the trenches that where all the battlefield casualties are left behind, and then the population dips a little bit at the interface. Now, what's really going on in the phase plane of in the phase plane of CA and CB, shown here, is that you're going out on a line that is very close to CA plus CB equals 1. And then you can look at the dynamics along this line. So the dynamics very rapidly takes you to this slightly bowed line, has an unstable fixed point. This is related to that double well potential that I was talking about. And if we now move on to discuss the dynamics of F, I've only talked about the dynamics of C, which is coupled to F. That's done on the next slide. I'll leave out all the algebra. But if you just look at what happens to F, along that line where CT is approximately 1, so I look along this, see if I can find this here. Laser pointer is back, and let me go now, hopefully, to the eraser. It's not letting me use the eraser for some reason, but don't worry about that. If I look along this line, CT equals 1, then the dynamics of F that comes from those two coupled equations, this setup here, is exactly what we talked about last time. The same third order polynomial we studied earlier if I rewrite epsilon A and epsilon B in terms of the selective advantage parameter delta and the antagonism parameter sigma. So
the concentration of A or B, but the relative concentration of one of them, let's say A, which will have the pulled or pushed wave. Is that right?
yeah, so, okay, good. Thank you. If I understand the question correctly, F is the concentration of A, okay?
Relative to a total, in like a, right?
So relative to C total. So in fact, if I go back here,
Yeah.
see F is CA divided by the sum. The sum is very close to 1. Small corrections that I can neglect. And so F is essentially the concentration of A, but it's the fractional concentration. And it turns out these two nonlinear equations approximately decouple, and I can treat the F equation by itself, and I get exactly what I had before. So the answer, the result, if sigma is positive, which it will be for antagonism, is that we will get back the pushed waves that I talked about in lecture 2. And then you can put in the fluid flow as well and look at what happens to F then, and then you get this very nice generalization to fluid flow of the F equation, the one that had the genetic wave, if U were 0. And what this is saying is that in a little fluid packet, advecting along at velocity U, F changes due to local diffusion, and it changes due to this third order polynomial in F, which, as we've seen, can be reinterpreted as a double well potential. So this is a deterministic general, there's no noise yet, generalization of model A to include the flow. Does that, is that helpful?
Yeah, thank you.
You're welcome. Okay, so I want to just show you some tests of that theory, since we showed that these reaction diffusion equations can at least be approximately modeled by a pushed wave. I want to check the predictions. And what are the predictions? Well, the one prediction is that if I have a little droplet of, say, a selectively favorite A phase, which wants to have a pushed wave, its dynamics, I didn't prove this last time, but I quickly showed some algebra that shows you how you might prove it. There's a critical radius required. And so here's the equation of how the droplet radius grows. And if the droplet radius is very tiny, okay, if the droplet radius turns out to be tiny, then maybe I would neglect this term here. And as a result, I have a droplet that is going to die. It'll wink out of existence. Whether this term dominates or this term dominates is determined by a critical droplet radius, which you can think of as the ratio of a line tension to a condensation energy. And it depends on the selective advantage and on the diffusion constant and on sigma. But if this is tiny, much smaller than RC, then this goes, you can solve this differential equation very easily, and R of t goes to 0 with this characteristic behavior here, kind of square root vanishing. However, if the droplet size is larger than RC, so if I go to the opposite limit here, so let me go to this eraser here. And if I go to the opposite limit where I get rid of, for R is large, I get rid of this term, then dr dt is just a constant velocity. And so R grows linearly with time, and we have the kind of pushed wave that I was talking about earlier. So we have both things are here. and we can ask, is this really correct for the coupled reaction diffusion equations that I was talking about? And so I'll show you the results of simulations of precisely these equations up here. For initially without flow, then I'll show them with flow in a minute, and we'll see whether these predictions are correct. So here's something that Zhao did, solving those coupled equations, where I have an initial droplet of a certain radius, which is less than this critical radius. It has a selective advantage, but it dies out because of the antagonism at the frontier. Here it is dying out. On the other hand, if I repeat the simulation with otherwise identical parameters, but with a larger initial droplet, it's larger than this critical radius. And in fact, we can estimate the critical radius, and it agrees pretty well with the simulation of the full coupled two nonlinear reaction diffusion problems, and it grows and grows and grows and takes over the system. So this is what Darwin would expect to always happen. It doesn't always happen because of the antagonism at the frontier. Sometimes it dies out. Okay, now I want to add some flow. So I'm going to look at the same kind of problem. This is a droplet that would shrink to 0 in the absence of flow. And we now put a saddle flow on. So this is an incompressible flow. And you can see there's a vortex going around here. There's periodic boundary conditions in the cellular flow. And there's a saddle point where the droplet will be compressed in this dimension and stretched in this dimension. So let's take a look and see what happens in that case. So it goes away just as it would in the absence of flow, but the flow crushes it. And so this one is really below the critical radius size. And in fact, the droplet goes away faster. If you're a droplet and you're born on a saddle, you're going to die more quickly than you would otherwise. Of course, there's an interesting question, what happens for a saddle flow which is greater than the critical radius? I'll show you that a little later. Here's a somewhat stronger, this is a smaller initial radius, and similar things happen except it dies much more rapidly. The driving force is 10 times bigger. That's what made it die so rapidly. But notice that as it dies, it splits. And we have a droplet that divides into two droplets before they both die. And the dynamics of this splitting that's going on here is controlled by a line tension that's controlled by the sigma factor. And if you systematically vary the strength of F, you find this characteristic dying of the droplet. Here's like 0F out here. The droplet dies like a square root until it gets rounded off, and this foot is a function of time. This is not the radius of the droplet, but something equivalent, which is the concentration here. And then as I increase F, it dies off faster and faster and faster. But it's a much richer system because if I make the droplet big enough so that it's greater than the critical radius and sensitive to these periodic boundary conditions associated with my cellular flow, I put on that same saddle flow. Watch what happens. The droplets merge again by virtue of these periodic boundary conditions. So the saddle flow is stretching it this way, squishing it this way. There's a line tension. It wants to minimize this line perimeter, this perimeter here. And this is the total concentration of A, which oscillates because of the periodic boundary conditions. But eventually, the selectively favored species, despite the shear flow, takes over and oscillates its way to dominance So the red is where the A species is, and after some of these oscillations, it takes over. So we just wrote a review article, which we submitted to Progress in Physics or some journal like that. I mentioned it at the beginning, and it was just posted on the archive a month ago. That's work with Suraj Shankar and some of my other collaborators. But I now want to conclude my lectures here in the last 20 minutes with actual laboratory experiments. And so I mentioned that, it would be nice to have laboratory experiments. Even low Reynolds number ones would be interesting. And that's this work, which we call, sometimes we call it On Growth and Form of Microorganisms on Liquid Substrates. And here's a more technical summary of our conclusions. And here are some of the results that we get when we have a flow. We thought we would make pumps and syringes in the laboratory in a liquid media to let these organisms move around. But in fact, we didn't need to do that, as I'll show you. But here are 3 different regimes, which I'll be talking about in the remaining 20 minutes or 15 minutes of range expansions. And you can see, depending on the viscosity of the fluid on which these yeast are floating. So they're basically floating at a liquid, nutrient-rich broth interface with air. And the dynamics is very different. And they generate their own fluid flow, even though these yeasts don't have flagella, they don't have cilia, there's nothing to make them locomote a priori, but they still create a flow, and the flow basically rips the colonies apart. And these are the people who really did the work here. Severine Addis is now on to her second postdoc at University of Chicago. Brian Weinstein was a grad student who worked with us, and this is my, as I said, my longtime collaborator, Andrew Murray. So, what's going on? Well, this is one of these yeast experiments, a little bit like the one I showed you for mutualists, but these are neutral. This is the homeland. This is the regime where the yellow and red strains are fighting it out. Here they localize into these sector-like spikes on hard agar. That's what happens on hard agar. We thought we would need, when we had a liquid, to put in pumps and syringes, but in fact, we didn't need to do that. They generated their own flow, and I'll show you what happens in detail. But if you now, this is a medium on which the yeast is floating that has a shear viscosity of 600 Pascal seconds. That's a finite 3D shear viscosity, but it's a very large one. Water, for reference, has a shear viscosity of 10 to the minus 3 Pascal seconds. So this is 600,000 times stronger, larger viscosity than water, but it's still viscous, and there's still flows. They're slow flows, but they're still there. And what actually explains this, a lot of what I'm going to tell you, I have to tell you why this happens. We figured this out by tracking the flow using fluorescent beads to figure out what's happening. So these green beads are not cells; they're just little markers that we can easily see under the microscope, and so we can map out the flow. What we found was that underneath the colony was a submerged vortex ring. And so in this axial cross section, you can see the fluid mechanics. The colony itself generates flows that stretch and dilate the mass they dilate and stretch the range expansion. So in fact, that's what we think is causing this epic of genetic demixing that gets stretched out here. So if, in a sense, this little annulus that would occur on hard agar becomes this much bigger annulus because there's this submerged vortex ring, which I still have to explain, but that's one aspect of these experiments. And you can sort of see it in this situation here, where in fact, I'll show you this range expansion. So let's just look at it. So you see there's complicated features here in the homeland. It's still not genetically demixing yet into sectors. It's being stretched by the flow. And as I keep going, you can see eventually I do get some genetic demixing out here. It chooses either to be a blue sector or a yellow sector. But also of interest are these features back in the homeland, the texture in the homeland. And if you look inside this red circle, there's a little feature which looked to us like a lizard, or an alligator, crocodile with a tail coming out here. So let's call it a lizard. And if you follow this lizard back in time, you can see that its shape is preserved. All the angles are preserved. The lizard gets smaller, but basically the range expansion, at least in this regime, is a kind of conformal transformation on the texture that was created at early times. And the lizard is just growing. It's like you painted the lizard on the surface of an expanding balloon, and it grows up to this size. And that part of the experiment, we think we can understand. The way we understand it is to imagine we have this colony that's growing, and we just think of the colony itself as a kind of 2D fluid. And of course, it obeys the equation of continuity, the 2D colony density. This is the density of yeast cells in this colony, obeys this divergence equation. And if the cells were not reproducing, the right-hand side of this equation would be zero, just conservation of cells. That's what this is saying in terms of some fluid flow. But in fact, there's a non-zero term on the right-hand side because the cells are dividing. And so there's a source term in this equation. And you can easily show that this implies that the divergence of this 2D velocity field, which is coming about because the cells are growing and pushing each other outward due to an excluded volume interaction, a kind of incompressibility, they're pushing each other outwards, that the divergence of V is non-zero, and we'll call that alpha 1. So here again, we have an advecting fluid with a non-zero divergence. And you can work out what the velocity field has to be. And it's a radial velocity field that is zero at the center of the colony and increases linearly with R as I go out. The colonies have a finite thickness. It's about 200 microns. That's about 50 cell widths. And they can't get too thick because they can't get enough nutrients up there to have the cell divisions. And this, we think that this radial flow accounts for how this strange lizard shape grows with time. You can work that out, and it seems to match. But that's not the only thing that's happening. In fact, you can look at the flow in more detail using these fluorescent velocimetry beads and map out what the flow field looks like. And it's not just non-zero underneath the colony, it stretches well out beyond the colony, maybe 40% further out than the colony. So this is the flow field that Severine Addis and Brian Weinstein mapped out. Here's the radial component of the velocity, which actually peaks at 30 or 40% of the colony radius and then slowly falls off. And this is characteristic, we think, that's how the flow looks, of a submerged vortex ring. I haven't told you why it should arise yet, but there's a submerged vortex ring, and it extends much further than the colony radius. And one way to think about it and get an estimate, for example, how things fall off here, is if this is the cross section of the vortex ring, then this is a region where there's a non-zero curl of the velocity field of the three-dimensional liquid gel here. Of course, there's a divergence of V equals 0 in the 3D liquid. And given my background, I find it convenient to map these equations onto magnetostatics because at least in the physics world, we have a famous equation, Ampere's law, which says that the curl of the B field is a current. So the current is sort of like this vorticity, and the velocity is like the B field, and then the divergence of B equals 0, and Maxwell's equations maps onto the divergence of B equals 0. So this, you can think of this as like two anti-Helmholtz coils describing the velocity field, and then there's an image vortex ring above the liquid-air interface to get the right boundary conditions. Anyway, that's how we understand it. But there's something going out here which is more vigorous than merely excluded volume interactions. But again, close in, it gives rise to this velocity field that goes out radially. And so a clue as to what could cause this very vigorous submerged vortex ring is shown here. When we first did these experiments, which showed like there was fluid flow going on and stuff, many of my distinguished colleagues in fluid mechanics said, "Oh, you're just careless experimenters. You have thermal gradients, for example, that you're just not controlling for, and you have Rayleigh-Benard convection or some experimental artifact." So we tried to prove them wrong, and I think we did. You can take away the colony and there's no flow. The colony is needed to cause the flow. But we also did other things. Instead of growing the colony at the liquid air interface, we grew it on the bottom just to sort of see what would happen. And we still got a submerged vortex ring in that case, but in this case, it compresses the colony rather than stretches it. But something even more interesting happened if we waited a long time. So these green dots are the PIV beads. This is the colony that you're seeing on the bottom of our Petri dish now. And you can see the green dots are moving a little bit. That's the beginnings of the flow. But something much more dramatic happens. The colony, turns out, is belching out gas. And in fact, if you look at how it metabolizes things in an approximately anaerobic environment at the bottom of this Petri dish, it takes glucose, or actually it's the dextrose handedness of glucose that it uses as a for food, and turns it into CO2 and ethanol. So when yeast does its work, it produces ethanol as a byproduct, hence it's used to make beer, for example. But in any event, it also makes CO2, and what we're seeing here is these CO2 bubbles coming out. And so that suggests that metabolism is going to be important. Now, this is an anaerobic metabolism that happens on the bottom of the liquid, but there's also metabolism that happens on the top. So let me return to that problem. And here is another metabolic pathway that takes D-glucose, otherwise known as dextrose, and also produces ethanol, produces ATP, it produces CO2 also, which goes off into the atmosphere. But what it does is it depletes nutrients just beneath the colony because it's sucking up the glucose this yeast media liquid, and it's sucking them up, and it makes the fluid underneath here, it turns out, a little bit lighter. In fact, we measured the density after the colony had done all the metabolism that it could. Density went down by 2 or 3%. And you can use this observation to solve the Navier-Stokes equations in what's called the Boussinesq approximation down here, allowing for a variable density, allowing for, now C is a concentration of nutrients that come into the colony with particular boundary conditions. And I won't go through the details, but the result, this is the colony edge, is a set of cross density and pressure contours. So green are the isobars of pressure, and the red are the isocores of density, and the pressure is just increasing as you go down, just like it increases when you go down at the bottom of a swimming pool. But the isocores of density are curved due to the colony shape, and the density gets higher as you go deeper into the fluid, because the nutrients are depleted and the glucose is slightly heavier than the average medium. So you have an increasing density going down and also an increasing pressure. And this turns out to be very important for the fluid mechanics, because when you have crossed contours of density and pressure, you could easily show that if you take the curl of the Navier-Stokes equations, that something interesting happens. So this is just the curl of the equation I showed you before. Let me go back and retrieve my pointer. This is the curl, the result of the curl of the Navier-Stokes equations. There are these nonlinear terms. Those were at so low Reynolds number that I can neglect those nonlinear terms. So here I will cross them out here, cross it out here, cross it out here. All that's left is this term here, and diffusion. So this is the vorticity, the curl of the velocity field. It's diffusing, but this is a kind of vortex mill, a vortex source. The gradient, you can see from these contours, the gradient of the density that looks like that. The gradient of the pressure looks like that, and they are at an angle. So their cross product is non-zero, and I generate a vorticity out of the plane here, which ultimately produces this submerged vortex ring. And in fact, you can take this cross product and convince yourself that the vorticity in cross-section will have this sign. There's an azimuthal symmetry. So this is the origin of that submerged vortex ring. So this is called a baroclinic instability. Basically what we did was to make a weather map of what's going on beneath our colony. And this causes weather patterns to move around also when you have gradients of in the atmosphere and gradients of pressure that are at different angles to each other. And I'll conclude by just showing you what happens to this system if I now increase or decrease the viscosity even further, thus limiting decrease the viscosity further. That lets you transport more, that changes the transport of this injected vortex ring, and new things happen. I'll skip this. So let's go over here and take a look at just a slightly lower viscosity range expansion than the one that produced that strange lizard. Looks completely normal up to now. But notice there are yeasts that are leaving the mothership. They are basically being pulled away from the mother colony by that submerged vortex ring. And if I drop the viscosity even more, if I drop it to 75% of what I had in the first video that I showed you, this is actually a 3-color range expansion. I get this, what we think is a very beautiful picture that to me, it looks like fireworks. I don't know if you shoot off fireworks on the New Year in India, but we have 3 different colors. We have yellow, blue, and pink. And you can see that these instabilities have an enormous effect on the population genetics. Each filament to first approximation is an individual color because of the initial sectoring. We still need to understand this better, but even the physics or the biology of these tendrils that are coming out in the fireworks display are interesting. And that's shown down here. Here's one of these tendrils, maybe this one coming off like that, or maybe this one. And it's growing. And then there's a kind of breakup into little beads, a kind of Rayleigh plateau instability, maybe. But it's not your conventional Rayleigh plateau instability of a liquid filament. These are alive. These are cells that are actively reproducing on the timescale of these experiments, which are like 6 hours. And 6 hours is great because that means the eddy turnover time, which is also about 6 hours, is very long compared to the doubling time of the yeast, which is in this case about 90 minutes. So the yeast have time, both genetically and from a population dynamics perspective, to adapt their behavior to what the very slow turnover of the submerged vortex ring is asking them to do. And so if I follow this movie out even further, you look in this gap, they're desperately trying to fill in the gap by reproduction, but it's not enough. And you still get this gap. So we still need to understand that better. Fireworks again. And then I'm almost done, which is good because it's almost half past the hour. Here's a model of what happens. Maybe I'll skip that. We think we can understand a little bit about this thinning process. But let me go to the last movie and go to an even lower viscosity. So I've now dropped the viscosity by a factor of 4. That means that the fluid motion, the vortex ring, is more vigorous, not damped out by viscosity quite as much. So it's a more vigorous submerged vortex ring. And we will see is a kind of viscoelastic response. Before we found that the colony is behaving like a liquid with these filaments and stuff coming out. But now it can't respond fast enough, and you'll see what happens. I don't know about you, but that reminds me of plate tectonics. This is like Pangea, and then the continents break up. This is like South America, this could be Africa. It behaves like a solid, which is ripped apart by this very vigorous submerged vortex ring. And the continents take over the entire plate in a day due to these self-generated velocity fields. And if you want to look more carefully and zoom in on the breakup of this initial primal continent, you can see the fracture mechanics is kind of interesting. It breaks up and forms these tendrils as it goes apart, but these are not like tendrils of a gel maybe that's being fractured, because the cells are actively reproducing, they're alive. And there's a lot of fluid mechanics and solid mechanics that needs to be done to better understand what's going on here. So eventually we get these continents, and if there was a submerged vortex ring underneath the initial continent, there's going to be a submerged vortex ring, we think, around each one of these continents. And you can see that these little fragments of yeast are lining up on the mid-planes between these continents. And we think that's precisely the downwellings associated with the submerged vortex rings, which the yeast resist. The yeast, over the timescale of our experiment, stay at the liquid-air interface. So we've sort of come full circle. It's not really the ocean, but it's certainly something kind of interesting. And I hope I've convinced you that with these accumulation lines for yeast, we have a system that's well worth further study. And with that, I will close and thank you for being so patient. Thank you for asking all the questions over my 4.5 hours of lecture. Happy to take more questions now. Thank you. Sorry to run over a little bit.
Thank you, David. Any questions? I think there's a lot we have to absorb. So
Exactly.
some time to think about it.
Maybe.
Yeah. But thanks so much, David, and for these lectures. And in spite of all these problems, you could make it and join. And I just wish that times were better and we could have you here in person and have full set of 5 lectures. But I hope there's another time. So thanks so much.
The videos were amazing, by the way, the plate tectonics and the alligator and everything. It's really cool.
Thank you. And I'm glad we can do this despite COVID. And I look forward to seeing some of you in the future in person.
Thank you. Bye-bye.
Right. Take care. Bye-bye.
Bye.